Hi, everyone. Good afternoon.
Hello.
Hello.
Hi.
Everybody doing okay?
Yep.
Yep.
Professor, I'm going to be joining in on my phone. I'm having laptop issues, so they're trying to work on it now.
Okay, Elena, thank you.
Yep.
Okay, so I'm just waiting just a moment to try to get a couple more folks online here. Okay. All right, so looks like one of our classmates is having a hard time getting logged in.
I was wondering if you might be able to tell me what my grade was from last week because I wasn't here on Thursday.
For your lab activity, Tiffany?
Yes, sir.
Okay. Sure, just a moment. Okay, so Tiffany, the grade I'm going to send you is for, let's see. For the termite one, that's the one you're requesting, Tiffany?
Yeah, was that the one that you gave everybody their grade for last week?
Yes. Okay.
Yes, then that's the one I
Here
was
it comes.
curious about.
Okay. All right. So Dallas, I'll send you yours, then we'll be starting content. Okay, hold on a sec. Okay, folks. Welcome back, everybody. Wow, having lots of internet issues today, it seems. Okay, so we will continue. I hope you had a nice weekend. I hope you're doing well. We're going to finish up our coverage of chapter 8 today and hopefully start chapter 13, which is the virus chapter. Okay, and I hope you've been enjoying doing the virus explorer lab activity. I think it's a really nice way to kind of introduce you to some familiar viruses that you've heard of. I mean, everybody's heard of influenza and, certainly coronavirus, things like that. So, okay. I just wanted to share, I want to share my screen and see if you folks have seen this piece of news. Okay, there's a news article in the news about, unfortunately, a tragic outcome associated with the microbiology topic. Okay, and here it is. Okay. Parents sue in boy's death from brain-eating amoeba. This is not a funny storyline. This is a real deal. Unfortunately, a 3-year-old boy passed away after playing around in a Texas splash pad. Okay. His parents are suing the city because it came to light that the people who own and operate, or whoever's responsible for maintaining the splash pad, did not keep correct levels of chlorine in the water. And so unfortunately, this beautiful little 3-year-old boy had an amoeba likely access his sinuses up here in the nose. That amoeba crawled in across the blood-brain barrier, got into the central nervous system, the brain. And once that starts, the amoeba starts to consume the brain matter. It actually eats the brain as food. And then the organism reproduces. So you get 1 amoeba, 2 amoeba, 4 amoeba, 8 amoeba, 16 amoeba, and all of a sudden you have an infection that is uncontrollable. Okay, there's no medications to deal with this. By the time you understand the reality of what is taking place, there's almost never a way to save a person from this, and it's a horrible outcome. The organism in play here is this amoeba, Naegleria fowleri. Let me open this up because the news article was nice enough to link information from the CDC about it. So this is what this microorganism looks like. Okay, and unfortunately, it did kill a beautiful 3-year-old little boy in Texas. So, you know, we typically only hear the sensational news stories when it's something bad like this, right? But that's the reality of it, that sometimes really nasty things, unfortunately, do happen. Let me just ask you in the chat window. Did you hear of this news story or any like it in the past? It makes you sit up if you're a parent. I mean, obviously, an adult could contract this as well. An adult could contract this, and it would most likely be lethal even to an adult. It would just take a little bit longer to die. So it doesn't matter whether it's a child or an adult, but you never like to hear about a child being killed because they were playing at a water park. I mean, that just is infuriating. Yasmin, what are the symptoms? Well, typically the child experiences headache and then becomes incoherent, like they can't walk, they can't talk, that kind of thing. So you rush them to the emergency room, and all of a sudden you realize there's massive swelling of the brain. So you do a spinal tap. You draw cerebrospinal fluid out of the child or the patient's body. You look at it under the microscope, and what do you see under the microscope? This is what you see right here, this microorganism floating in the cerebrospinal fluid. So it's the worst nightmare of any person, particularly a parent. Kit, you say there was that flesh-eating bacteria outbreak at one of the beaches, wasn't there? Yeah, there's been many cases of flesh-eating bacteria at beaches all around the world. I'm not aware of that one in Florida at the Florida Keys, Kit, but I'm sure you're right. There's been flesh-eating bacteria down at Ocean City, Maryland. Okay, Atlantic City. There have been cases. Okay, never go into that kind of water if you have a cut on your leg. Don't do it. You're asking for trouble. Okay. All right, so I just wanted to share with you, unfortunately, a sad story, but to show you that, you know, microbiology does impact our lives every day, and we need to be aware of this. Okay, gang, I gave you some homework here to take this DNA sequence and transcribe it into RNA, and then take the RNA and translate that into amino acids. So please type into the chat window what you got for the RNA sequence. What did you get for the RNA sequence? Okay. What did you get for the RNA sequence? Okay, and so as you finish entering your information, you know, just take a look. Just take a look in the chat window to see if you agree with your classmates. Okay, and it looks like most people do agree. It looks like most people do agree. There might be one or two that has a slightly different thing. But I will say it looks like the majority of you got it right. And so let me type this in here. Okay, let me say this. And, you know, I have to say it, gang, because I'm your teacher. So I've got to say the things you don't want to hear. If you gave me this sequence on an exam and you did not specify the directionality of the molecule with the 5' and 3' business, you would lose credit. You would lose credit, okay? So the actual nucleotide sequence, most of you seem to have gotten correct. And it is in fact AUG, CCU, UAG, CAG, 5'. Nicely done, okay? So make sure you're paying attention to the directionality of the molecule that is posed to you. I mean, I don't mind telling you, you're going to have something like this on the next exam. It's going to come at you, okay? So you need to start thinking about it. All right. So I have a question for you in the chat window here. Here we go. When RNA polymerase made this piece of RNA, did it physically bind to the 5' end of the DNA and move to the right? Or did it physically attach to the 3' end of the DNA and move to the left? Okay, I'll let you read that and let you soak it in, and then give me your best answer. Okay, we're starting to get some answers up here. And I appreciate that, I really do. Okay, give another few seconds. Anybody wants to put in an answer? Okay, thanks everybody for contributing your responses today. And drum roll, you are all incorrect. That is wrong. Okay, that is wrong. RNA polymerase would have sat down on the 3' end of the DNA and moved to the left. Okay, so let's talk about this before we go any further. During transcription, RNA polymerase always synthesizes new RNA 5' to 3'. Okay, let's take a look at that. Kit, the quick answer to your question is no. So let's take a look at this statement that I'm highlighting in your notes. During transcription, RNA polymerase always synthesizes new RNA 5' to 3'. So what I'm telling you is, for this piece of RNA to have 5' on the right of your view screen as you're looking at your computer, RNA polymerase would have made the RNA moving right to left, which means it had to attach to the 3' end to do the 3' end of the DNA to do that. Okay, so let me just continue here. During transcription, RNA polymerase always synthesizes new RNA 5' to 3'. This means RNA polymerase sits down on the DNA template on the 3' end. Okay. So let me just ask you, because I don't want to lose you, is this making sense? Does this statement that I put up in bold, well, capital letters, because it's really, really important, is it making sense to you? Okay. Folks, what you need to do is take your humanness out of this topic. Take your humanity out of it. And what I mean by that is the following. Since you were all raised, I honestly don't know your background, but I'm going to take a guess that the majority of you were raised probably in the United States. And we teach children in the United States to read books and articles, magazines, newspapers. We always teach you to read left to right across the screen, left to right across the page. And so in our minds as humans, we get to think that the machinery of the cell has to move left to right. No, 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 the machinery doesn't care two poops about left and right. It doesn't care about left and right. It cares about the directionality of the molecule. RNA polymerase will always make RNA in a 5' to 3' direction. So to achieve that goal, it always sits down on the 3' end of the DNA. And whether that 3' end is represented on a piece of paper on the right or left side of the page does not matter. It does not matter at all. Okay, so let me say this. All known polymerase enzymes, DNA and RNA polymerases, always synthesize new nucleic acid 5' to 3'. There has never been an exception that has been found. Maybe someday we'll find it. Maybe by the missing link of life, who knows? But all known polymerase enzymes, whether they be of the DNA or RNA polymerase variety, synthesize their new nucleic acid 5' to 3'. Okay. Any questions about that? All right, folks, let's go for broke here. Let's go for broke, and I'm going to change my coloring here. I'm going to go with a nice blue here. Okay. Hate computers. Okay, folks, type in your sequence of amino acids into the chat window. What did you get for your amino acid sequence? Okay, I'm just going to give you folks a few more seconds so more people can contribute answers here. Okay, let's get at least 2 more people contributing in a response before we continue. Okay. Okay. All right. So if anybody else wants to contribute, go right ahead. Okay, folks. Taylor, I see your message. Thank you. Take care. See you later. Okay, so folks, drum roll. You are all incorrect. Everything in the chat window is wrong. Okay? Everything in the chat window is wrong. So I'm not going to give you the answer on this. Redo this for next time. Okay? You are wrong. Redo it. Okay? So let me give you a hint. Let me give you a hint here. Hold on. As your instructor's desk absolutely falls apart on him. Okay, hold on a second. So I'm opening up the textbook at this moment, and I'm trying to locate a page that gives you a hint about this, okay? All right, so here's my hint in the chat window. Here's my hint in the chat window. Here we go. Hint for translation. Information near figure 8.8 will tell you how to translate properly, okay? So I'm going to ask you to revisit that over the next 2 days. Okay, please revisit that over the next 2 days because nobody got it right. Okay. All right, gang. So, you know, if I'm truthful here, I would have bet money that you didn't get it right because students don't focus on the things they need to focus on. But hey, that's why you're students, right? Okay, so try again. I want you to try again. All right. We need to finish the lactose operon. That's what we need to do here. Okay. So you remember our conversation from last time. We spent a good bit of time diagramming out in a schematic form the, let me see if I even have it quickly accessible. Yes, right here. Okay, in schematic form last time, we drew out the schematic of the lactose operon here that's now on your screen. And we went through and we defined everything. Okay, let me just read these things quickly. Lactose repressor operon, P is the promoter. LAC-I is the gene that codes for the lactose repressor protein. That gene is always on, right? The LAC-I gene's always on. In terms of the lactose operon, we have the cap site up here. The cap site is actually a part of the promoter, and it is a sequence of DNA that in some cases will be bound by a protein called cap. P is the promoter. O is an operator. The operator is a DNA word that is the binding target of the lactose repressor protein. Z is a gene that codes for an enzyme called beta-galactosidase. Its job is to break down the sugar lactose. Y is a gene that codes for an enzyme called permease. And the job of the permease enzyme is to bring the sugar into the cell. A is a gene that codes for an enzyme that we truly don't fully understand, but we have an educated guess in science that this enzyme might help detoxify the environment from chemicals that are produced during the lactose metabolism process that are kind of toxic to the cell. That's our guess right now. We might be wrong. Okay, so that's what we put in last time. Before I continue, do you have any questions about that? Okay, so let's show you a video. Let's show you a video on the lactose operon. I actually have two videos here. They're both on the lactose operon. I'm only going to show one in class. The other you can watch on your private study time. So let me do that. I want to show the first video. Okay. Okay. So let me get this.
If you're learning guitar late in life, there's one distinction that will determine if you're going to succeed or fail. And I'm going to show it to you in this video.
Escherichia coli, 2 regulatory.
I'm going to start it from the beginning. Here we go.
In Escherichia coli, 2 regulatory sites, the cap binding site and the operator site, function together to ensure that the 3 proteins required for growth on lactose are produced only when glucose, a more efficient substrate, is absent and lactose is present. The cap site is a region of DNA adjacent to the promoter. In the absence of glucose, a high level of cyclic AMP, cAMP, builds up in the cell. The cAMP binds to the catabolite activator protein, cap, and activates it. The cap, with bound cAMP, can now bind to the cap site and bend the DNA, allowing RNA polymerase to associate with the promoter. When glucose is present, cAMP levels are low, and the cAMP does not bind to the cap. The unactivated cap is unable to bind to the DNA, and RNA polymerase does not associate with the LAC promoter. There is a second regulatory protein called the LAC repressor. This protein is capable of binding to the operator site and preventing transcription when lactose is absent and the 3 proteins of the LAC operon are not needed. The repressor covers part of the promoter when it binds to the operator, preventing RNA polymerase from making messenger RNA. When lactose is present, a lactose isomer binds to the repressor and inactivates it. This prevents the repressor from binding to the operator site. RNA polymerase can then associate with the promoter and carry out transcription. Transcription of the LAC genes is said to be induced by lactose.
Okay, gang. So there in 2 minutes of molecular chaos, you have a brief description of the regulation of gene expression in the lactose operon. And I would encourage you to definitely watch this a few more times during your private study time to see if you get it, okay? It's one of those things that's involved enough that one pass through is usually not enough. But let me just ask you while I get set up to take notes, do you have any questions about the video?
Not about the video, but would you mind scrolling up? Last time I didn't get 5 and 6 under the lactose operon, and I would just like to jot those down quick, please.
Oh, on the notes.
Yes.
Okay. Did you get the notes in email?
Oh, you're right. I did. So I have those. Thank you.
Okay, sure thing. All right. Okay, gang, let's see if we can make heads or tails of the lactose operon. Okay. Okay, I'm hoping to take no more than about 20 to 30 minutes on this, and then we can start the virus chapter. Let's see how it goes. Okay. So question I'll put it in your notes, okay? And I'm going to ask you to give me some feedback in the chat window, okay? Under what circumstance would you expect the genes of the lactose operon to be turned on, which is to say, to be expressed? Give me your response in the chat window. Under what circumstances do you expect the genes of the lactose operon to be turned on? Okay. Thanks for your contributions, Elena and Chelsea. And folks, take a look at the chat window. You see that Elena and Chelsea disagree. They disagree. So maybe we should have them do an arm wrestling contest. That would be interesting. Okay. All right. So we're getting a lot of things in here. Okay. And I know some of your answers are coming out from the video you just saw. Okay. But you know what? Of all the answers, I really like Fatimata's the best right now. Under what circumstance would you expect the genes of the lactose operon to be turned on? It's when there is lactose present in the environment, right? In other words, when there is lactose sugar present in the environment that could be utilized as an energy source, that's the exact moment we would want the genes of the lactose operon to be turned on so that we can eat and extract energy out of the lactose sugar. Okay. So that's the first answer, when lactose sugar is present in the environment. And think about it. Does it make any sense to spend cellular energy to make lactose eating enzymes if there is no lactose to eat? Think about that. If there is no lactose in the environment to eat, why should a cell spend any of its energy to make enzymes to eat something which is not available? Makes sense, doesn't it? Why should I spend time preparing a recipe when I know that one of the key ingredients to that recipe is not in my refrigerator? That seems like a horrible waste of time. Okay. So the first answer is, what the heck? Oh, sorry. Under what circumstance would you expect the genes of the lactose operon to be turned on? It's when the lactose sugar is present in the environment. that hopefully makes sense to you. Okay, next, you now learn from the video that there is a second requirement. So let's do it this way. Requirement 1. And secondly, there's a second requirement that the video shared with you, and that was when glucose, which is an alternative sugar, is not present in the environment, or at least is at very low concentrations, okay? When glucose is absent from the environment. This is requirement 2. Okay, so there are actually 2 requirements to get the lactose operon genes to be turned on. Number 1, there's no glucose in the environment, and number 2, there is lactose present in the environment. What the heck is going on here with this glucose business? Okay, so question, why should a bacterial cell like E. coli care if glucose is present when all it wants to do is eat lactose? And here's the answer, folks. Bacteria, which are cells, like any cell on the planet Earth, whether they're bacterial cells or the animal cells making up your body, Mother Nature has created cells with a preference for what sugars will be used first before other sugars are used. Mother Nature has endowed cells with a hierarchy of which sugars should be eaten first, which sugars should be broken down first, which sugars should be metabolized first, okay? Mother Nature has evolved a hierarchy of sugar utilization, okay? The most preferred sugar for all cells to metabolize is glucose. If you've had any other biology and any other courses, maybe you have at some moment in time studied those horrible things that students hate called glycolysis, Krebs cycle, electron transport chain, okay? Usually not a fan favorite for students, but they are deeply important for how cells function on our planet. So this hierarchy of sugar utilization is actually being given a name, and the phenomenon of regulating the sugar usage hierarchy is called catabolite repression. Catabolite repression refers to the phenomenon when the presence of one sugar when the presence of one sugar prevents a second sugar from being metabolized. This is the phenomenon observed when the presence of one sugar interferes with the metabolism of a second sugar. Okay, now in terms of sugars on our planet, the science has come across, science has been studying this for some decades now, and we have a little bit of a list of what sugars get eaten and used before other sugars and so on and so forth. I'm not going to give you that list, but from studying the lactose operon here and seeing the video, it should be obvious to you that if cells have glucose available, they will preferentially eat and use that first before they do anything else. So if glucose is present in the environment, we're not going to turn on the genes to eat lactose because the cell doesn't want to eat lactose if glucose is available, okay? It's kind of like in our human experience. If you're offered 2 plates of food, one plate of food has a freshly grilled steak, garlic mashed potatoes, country green beans, and the other has, I don't know, lima beans, and some other liver, who knows? Okay, you're going to have a preference. You're going to have a preference. Thanks, Kit. Brussels sprouts, right? Not my favorite either. You are going to have a preference for which plate of food you eat, right? Because you have a preference. You know what tastes good and what doesn't taste as good. Would you eat the plate of food that didn't taste good if that was the only food available and your survival depended on it? You bet you would. Everybody would. Yes, I'll eat the Brussels sprouts if I'm going to die because I don't have any food. But otherwise, I'm eating the steak and the garlic mashed potatoes because that tastes a lot better. Now, I don't know that bacteria have a sense of taste. Okay, they don't have taste buds, but they do have a biochemical and energetic preference for which sugars. In other words, typically bacteria and cells can get more energy from breaking down glucose than they could from breaking down lactose. And so they'll preferentially use the glucose. Okay. All right. So here's the regulatory scheme in a nutshell. Any questions you'd like to ask? Okay. Let me put up the schematic again for the operon so we can keep it in our vision here when we're thinking about this. Here's the schematic one more time. I'm just going to put it in like this: PI cap POCYA. Okay, we drew it out last time. Here it is in okay. Paula Shadi, I see your comment and I understand. Okay, all right. All right, so here we go. Okay, so we know from our notes last time that the lactose repressor protein is always made because the lac repressor gene is constitutive. Because the lactose repressor gene, which is gene I in the schematic, is constitutive. Okay, so what that means is that the repressor protein is always made, and the repressor protein has a binding target. Can you tell me in the chat window what the binding target of the repressor protein is? What does lac repressor bind to? Thanks, folks. I really appreciate those that are, staying focused here because this is a difficult topic. You did a great job. Yeah, it absolutely binds to the operator. So the repressor protein binds to the operator. And what that means is the repressor protein is capable of physically blocking any RNA polymerase molecule that wants to gain access to genes Z, Y, and A. Okay? The lac repressor protein binds to the operator section of the lactose operon promoter. This blocks RNA polymerase from accessing genes Z, Y, and A. Okay, what I'm saying is if lactose repressor is bound to the operator, the lactose operon genes are shut off. Okay. If lac repressor is bound to the operator, the lactose operon genes are off. Okay. Okay. So here's a question. What would get the repressor protein to detach from the operator? Okay, what would get the repressor protein to detach from the operator? Anybody know the answer to that? Okay, so I'm just going to run with Kit's response there. Okay, Kit says lactose, and Kit is absolutely right. If lactose sugar is available in the environment, it will bind to the repressor protein, change the shape of the repressor protein so it can no longer attach and stay connected to the operator, and the repressor protein and lactose complex falls away from the DNA, and now RNA polymerase can go right through and transcribe genes Z, Y, and A. Lactose. Okay, so here's what happens. Okay. When lactose is available, it binds to the lactose repressor protein, changes its shape, causing the complex of lactose repressor protein to detach from the DNA. This exposes the genes of the lactose operon to RNA polymerase activity. Okay, so let me pause for a moment and read my notes to make sure it's right. Then I'm going to read your comments in the chat window. Okay, when lactose is available, it binds to the lactose repressor protein, changes its shape, causing the complex of lactose and repressor protein to detach from the DNA. This exposes the genes of the lactose operon to RNA polymerase activity. Yes, that is a correct statement I put in the notes. So now on to the chat window. Chelsea asks, can you have an instance when something other than lactose binds to the repressor protein and changes the shape so lactose can't bind? Yes, Chelsea, you can. There's a molecule that scientists have discovered by dumb luck, which is known as a gratuitous inducer of the lactose operon. Maybe you've heard of it. It's called IPTG, isopropylthiogalactoside. IPTG. So yes, Chelsea, that can happen, although most times it doesn't happen. Kit's question, it just seems weird that you would need to use up lactose to stop the repressor protein when the whole point is to make enzymes to digest lactose. It's like you're using up the lactose to make the stuff that eats the lactose. I understand your position, Kit, and let's see if we can get further into the conversation. It might be clearer what the logic of Mother Nature is here, because let me give you the punchline. Once we remove the repressor from the operator, gene Z gets made, which is beta-galactosidase. So gene Z makes an enzyme. That enzyme goes up and it nibbles on the lactose that's attached to the repressor, and the cell gets energy from breaking down that lactose. Because that lactose has now been metabolized to produce energy, that lactose is no longer bound to the repressor. Since it's no longer bound to the repressor, the repressor changes back to its original shape, reattaches to the operator because there's no more lactose around. So we shut down gene expression of the Z, Y, and A genes a second time. Okay. Wow, right? I mean, it's kind of like, holy crap, who figured this out? Nobody figured it out in nobody figured it out on a Tuesday afternoon, folks. Talk about decades of research, decades. Okay. So there's another piece to the puzzle here. Okay, so this explains how lactose, maybe I should put some words in your notes about what I just responded to Kit about, okay? When lactose is attached to the repressor, LAC Z, Y, and A genes are turned on, and beta-galactosidase, the product of the Z gene, begins to metabolize, meaning it begins to break down the lactose that is free floating inside the cell and lactose that is attached to the repressor protein. Okay, when the lactose attached to the repressor protein is broken down, the repressor protein changes its shape back to a form that allows it to reattach to the operator DNA, effectively turning off the expression of the lac operon genes. Okay, so folks, the notes you have at this moment in time explain the lactose turning on and turning off, does not yet explain glucose, and that's our next piece of the conversation. But do you have any questions with what I presented so far? This is a lot to process, is it not? A lot to process. Okay, so if lactose is present, it binds to the repressor. The repressor detaches from the operator. RNA polymerase can bind to the promoter and go across and transcribe genes Z, Y, and A. When RNA polymerase does that, you get LAC-Z messenger RNA. The LAC-Z messenger RNA is subsequently translated into beta-galactosidase enzymes by the activity of the ribosomes in the cell. That enzyme then starts nibbling on the lactose like a little molecular Pac-Man. If it nibbles on the lactose that's attached to a repressor, then the repressor changes shape back to its original form and reattaches to the operator. And then you block any new RNA polymerase from coming through. So we turn gene expression back off. Now we got to bring in, we've kind of approached this backward, but it's okay. Let's talk about glucose, okay? So we got to talk about this darn cap binding site up here. Okay, we got to talk about that, okay? All right, so let me just put a question here. How does glucose play a role in regulating expression of lactose metabolizing genes? Here's how it's done, okay? There is a cap DNA sequence that's in the promoter of the lactose operon. That's what you see in red in the schematic above. There is also a gene that we haven't told you about called the cap protein gene that's at another location of the E. coli chromosome, and it is a constitutive gene, and the cap protein is always manufactured, okay? So cap is confusing because it refers to two things. First, there is a section of the lac operon promoter, which is the cap binding site. Okay, maybe that would help if I put that in this way. Cap binding site. There is a gene that is used to make cap protein. It is constitutive. It is always on. Okay, it is always on. Now, by itself, cap protein cannot bind to the cap binding site. Okay, let me ask you a question in the chat window, and it's okay if you can't answer, but I want you to try. I'm almost there, gang. Hold on here. Here we go. What did the video tell you about cap protein binding? What must be attached to the cap protein for it to bind to the cap binding site? What must be present? Did you pick up on that? And it's okay if you didn't, because this was a lot very quickly. Anybody? Okay. If you really want to look at this, you would be looking at figure 815, figure 815. Take a look at figure 815 in your textbook. So I'm going to put that into your notes. Reference figure 815. Okay. It is cyclic AMP. Do you see figure 815 in your book? Do you see cyclic AMP there? CAMP. Okay. So by itself, cap protein cannot bind. It cannot bind to the cap binding site. Okay, so here we go. Cap protein can only bind to the cap binding site when it itself, meaning the cap protein can only bind to the cap binding site when it itself is bound by cyclic AMP. The story deepens. It gets even more confusing, right? So what the heck's going on with this cyclic AMP business? The production of cyclic AMP is directly tied to the presence and absence of glucose. The manufacturing of cyclic AMP is similar to a glucose sensor. Here's the deal, folks. When glucose is high, in the environment, when there is lots of glucose present, cyclic AMP levels are low. When glucose is high, cyclic AMP levels are low. When glucose is high, cyclic AMP is low. They're inverses. So here's the deal. If there's a lot of glucose, cyclic AMP is going to be low. If cyclic AMP is low, then there's no cyclic AMP to bind to the cap protein. And if there's no cyclic AMP to bind to the cap protein cannot bind to the cap binding site. And if the cap binding site is not attached with cyclic AMP cap protein, then the DNA does not get bound in such, it does not get kinked in such a way that allows RNA polymerase access to the promoter. Okay, let me type this out. Holy crap. All right, so here we go. Okay, if cyclic AMP is low, there is no cyclic AMP to bind cap protein. This means There is no cyclic AMP cap protein dimer to attach to the cap binding site of the lac operon promoter. Without this RNA, without this binding, without this dimer binding, the DNA is not bent in a manner that allows RNA polymerase to attach to the promoter. And effectively, this keeps the lac operon genes off. Okay? So even if the conditions of lactose being present are able to remove the repressor protein that we talked about up here in statements 1 and 2, even if that's true, if glucose is high, then you're never going to get RNA polymerase binding to the promoter in the first place, and you're still not going to express the LAC-Z, Y, and A genes. Okay. Questions? Type yes if you are passed out. Okay. All right. So I did see a question there from Kit. Kit says, so you always need glucose present to be able to produce lactose-eating enzymes. No, Kit, exactly the opposite. When glucose is low, cyclic AMP is high. Okay, let's put that into the notes. When glucose is low, cyclic AMP is high. If cyclic AMP is high, then cyclic AMP can attach to the cap protein, allowing the dimer to attach to the cap binding site. When the dimer binds to the cap binding site, it bends the DNA in a way that allows RNA polymerase access to the lactose operon promoter. Okay. All right. So let's look through that reasoning one more time here. Okay. When glucose is low, when glucose is not available in the environment, when it's not available, cell can't expect to use it for an energy source, right? Because it's not available. So if it's not available, the cell better find a second different energy source. Maybe lactose would fit the bill. So if glucose is low, cyclic AMP production goes up, so cyclic AMP levels get high. If cyclic AMP is high, it will attach to the cap protein. The cyclic AMP and cap protein as a dimer attached to the cap binding site that's part of the lactose operon promoter. When it does so, it actually kinks the DNA. It actually bends the DNA. We have X-ray crystallography data that prove this. So attachment of the cyclic AMP cap protein to the cap binding site DNA actually physically bends the DNA. And when you bend the DNA, you expose the promoter of the operon, the lactose operon, better so that RNA polymerase can sit down on it. And then at that point, as long as the lactose repressor protein is not in the way, because lactose is attached to the repressor and removed it, then the RNA polymerase can move merrily along and transcribe genes Z, Y, and A. Questions? I mean, it's kind of grotesque at how complicated this is, but at the same time, when you finally do get a glimmer of understanding of how this is working, it's kind of like, wow, this is amazing what Mother Nature has come up with. Mother Nature has created an amazing regulatory circuit which couples glucose metabolism and lactose metabolism. If glucose is present, we're going to shut down production of the lactose-eating enzymes. If glucose is absent, well, we're going to give production of the lactose-eating enzymes a fighting chance to take place. Okay, why, who told Mother Nature to make it complicated? Yeah, I don't know. Maybe it was Father Nature. I don't know. But at any rate, it is what it is. And let me say to you that the story that's just been told to you is the culmination of about 60 years of research, of understanding this. And yeah, Rachel, it's always Dad's fault. Yeah, I understand that because I'm a dad. I get blamed for lots of things. But at any rate, this story has been unfolding and research has been carried out for upwards of 60 years on this. There's still aspects of it we don't fully understand, but we'd have this one knocked down pretty good. This is one of the best understand genetic regulatory circuits in all of science. We understand it just about better than anything else, which is why it's always presented in textbooks, because it's the best understood. And truth be told, having a deep understanding of these topics has allowed scientists to do genetic engineering, to do things in cells that we never knew we could do before, and control the production of things in a very specific manner, using elements of the lactose operon to do that. Okay, so this has real relevance for genetic engineering and the production of molecules and substances we might use to treat people, treat patients. Okay, folks, I don't want to beat a dead horse. I would like to start chapter 13, but I'll pause right now and ask if you have any questions you might like to ask here about this system. Imagine if I did hold you responsible for the tryptophan operon in your textbook. I'm not, but imagine if I did, right? I mean, it's just insane. It's absolutely insane. Okay. All right, folks. We're going to put that to rest, and you study it a little bit and tell me how I can help you, okay? All right. Let's go to chapter 13. Chapter 13. I might as well put it on a brand new page. Let's do that. So chapter 13 is viruses. These are one of my favorite things to talk about. You know, we're all experiencing the pandemic because of a virus. It's so irritating. But when you can get past that kind of aspect, viruses are just really cool things, man. I mean, they are just amazing little things that Mother Nature has come up with. Okay. So you're doing a lab activity for me right now on viruses, and I hope you're enjoying that, even though it's work. So let's just start with a couple talking points here. Okay, so I'm on page. I know our numbering is a little different if you have an electronic book. I'm on page 362, somewhere around table 13.1 and figure 13.1. Okay. So first off, you know, let's answer the question, what are viruses? Okay, you probably can answer that now that you've been doing homework, but let me give you my take on it, okay? Viruses can be described as obligate intracellular parasites. Viruses are obligate intracellular parasites. And, you know, take that phrase, right? Obligate. Okay, so obligate means that the virus is required. It is required to be inside a host cell to replicate and do something. Otherwise, the virus is pretty much inert and unreactive for the most part outside of the cell. The virus can't reproduce outside of the cell. Viruses are required to be inside cells to reproduce. Intracellular, well, that's just telling you that the virus has got to be inside the cell. Viruses have their most meaningful metabolism inside cells. And of course, a parasite, well, it's something that kind of lives off something else, right? Kind of like your children are parasites for mom and dad, right? But it's a good parasitism in that case, I guess. All right. The microbe lives off another cell. Remember, viruses are not cells, they're viruses. Viruses have no plasma membrane, right? All these sorts of things. Some of the things I showed you in videos in lab last week, and maybe with your homework, it talks about whether viruses are alive or dead. Honestly, it's a philosophical debate. Rachel, I'm sorry, I just saw your note. Around page 362, 363, table 13.1, figure 13.1.
Thank you.
Whether viruses are alive or dead, it doesn't matter what the answer is, because obviously they do infect cells, they alter the metabolism of the cells, they make us sick at times, so we have to be concerned about them. So, some folks argue that viruses are alive and others argue they are dead. Well, who cares? Okay, they do make us sick. Okay, so we need to be aware of them and think about them because of that. So some other things that probably came up with your study time and reading and so forth. It's been estimated, and it's just an estimate, so take it with a grain of salt, but it's been estimated. Now, let me ask you a question in the chat window. Here we go. Have you ever gone swimming at the seashore and had some water splash into your mouth? So hopefully you have, right? I mean, you got to enjoy life. You got to enjoy some life, so go swimming. But here's the deal, okay? Estimates of the ocean water on our planet estimate that every one teaspoon of ocean water typically contains about 50 million virus particles, okay? So you just had a nice gigantic helping of virus particles, okay? But by and large, we don't get sick from these because these virus particles are virus particles that are for marine life, right? For fish. Fish viruses don't tend to infect human cells, right? So they're not that dangerous to us. You could have, some of the plant life underwater, right? Kelp and things like this, they have viruses too, but they're not going to infect a human cell, so you can largely consume them and they'll be destroyed by your body, pass through your body, will not cause you harm, right? So even though you just swallowed a whole boatload of virus particles, they were virus particles that were not designed to infect your cells. So that's generally not going to be harmful to you, okay? Number 4 here, okay? The video I showed you in lab last time mentioned to you that about 8% of the DNA sequence inside human cells can be traced to viruses that have that are in our DNA or have been in our ancestor cells a long time ago, and they kind of set up shop like a fossil. Okay, so estimates trace about 8% of our human DNA to viral activity. Okay, that's about 1/12 of our genome. You know, if I were to guess, I personally think the percentage is higher, and I think we will discover that it's going to be higher over time. But right now, 8% is still not an insignificant amount, okay? So the human genome, if we talk about the diploid genome, that's 6.4 billion nucleotide pairs of DNA. So a typical skin cell on the surface of your skin has 6.4 billion nucleotide pairs of DNA, right? That's 6.4, scientific notation 9. Now let's multiply that by 8% times 0.08. Okay, so about 512 million nucleotide pairs of DNA sequence inside your cells is somehow related to a virus. Okay, that's a pretty big number, isn't it? Pretty big number. Okay, so that may be of interest to you. And let me say this, I know I'm getting close on time. Our human DNA definitely has fossilized versions of virus that is part of our DNA sequence. And in fact, these DNA sequences are given a special name. They're known as HERVs, H-E-R-V-S, okay? Fossilized, and for the most part, dormant viruses that are part of our human DNA are called HERVS, just like that, which stands for human endogenous retroviruses. Human endogenous retroviruses, okay? Well, what the heck are these things? These HERVs that we have in our DNA, these are primitive viruses that at some part in the evolutionary history of our species, Homo sapiens, they infected our ancestors. So maybe your grandpa 10 generations ago, got infected with a virus, and grandpa's immune system had to deal with that virus. And one of the ways that grandpa's immune system might have dealt with that virus is to take a snapshot of the virus. Oh boy, you guys having trouble here? Okay, hold on. Okay, I just stopped sharing my screen. And I'm sorry, let me try to reshare and see if that wakes things back up. Okay, so I'm just going to scroll up and down. Is my screen moving up and down for you? Can you see me moving the notes up and down?
Yes.
Oh dear, not everybody can.
Mine's not moving at all.
Oh, I'm sorry, folks.
Yes, I can see it.
Okay. So some of you are okay and some of you aren't.
I can see it
It
as
was
well.
freezing earlier though.
So yeah, I'm sorry about that. I mean, you know, I can never tell whether it's my computer system or whether it's your computer system, whether it's my internet or your internet. So just do the best you can. Okay. So
Yeah, it's definitely lagging. I agree with Rachel.
okay. All right. So let's see. I will turn my video off for my, well, no, I can't do that. Hold on a sec, folks. If I do this, can you still see my notes? Can you see the notes?
Yes.
You can? Okay.
Yeah.
I'm gonna turn my video off. Is that better in terms of choppiness?
Yes.
Okay, all right. So I'll go with video off for right now. let's finish up the story, then I'll let you go, promise. These HERVs, okay, they are ancient viruses that infected our ancestors. And that our ancestors, as a collective group of human beings or a collective group of primates, if we go back far enough, the collective species immune system deactivated these viruses and left a fossilized copy of the virus in our DNA as sort of a record of infection that has happened in the past. So what I'm saying is these ancient viruses are kind of equivalent to HIV 5,000 years ago, right? 5,000 years ago, as far as we know, HIV did not exist, but there was some kind of virus a long time ago that was threatening to our ancestors, just like HIV is threatening to us today, okay? This was our ancestors' HIV that was eventually defeated by our species' collective immune system. Okay, so these deactivated viruses now show up in our current DNA, and we can see them when we do DNA analysis. And the point of it is that 10 generations from now, our great grandchildren, our great grandchildren, many generations from now, they are going to have viruses in their DNA that we right now are currently wrestling with, such as HIV, right? So our future grandchildren will have fossilized